The antimicrobial potential of the neem tree, Azadirachta indica. It's written by Marina Wiley and Scott Merrill, printed in Frontiers in Pharmacology. Neem, Azadirachta indica, also known as the neem tree, has been used for millennia as a traditional remedy for a multitude of human ailments. Also recognized around the world as a broad-spectrum pesticide and fertilizer, neem has applications in agriculture and beyond. Currently, the extensive antimicrobial activities are being explored through research in the fields of dentistry, food safety, bacteriology, mycology, virology, and parasitology. Herein, some of the most recent studies that demonstrate the potential of neem as a previously untapped source of novel therapeutics, and summarized as they relate to the aforementioned research topics. Additionally, the capacity of neem extracts and compounds to act against drug-resistant and biofilm-forming organisms, both of which represent large groups of pathogens for which there are limited treatment options, are highlighted. Updated information on the phytochemistry and safety of neem-derived products are discussed as well. Although there is a growing body of exciting evidence that supports the use as an antimicrobial, additional studies are clearly needed to determine the specific mechanisms of action, clinical efficacy, and in vivo safety of neem as a treatment for human pathogens and of interest. Moreover, the various ongoing studies and the diverse properties of neem discussed herein may serve as a guide for the discovery of new antimicrobials that may exist in other herbal panaceas across the globe. Medicinal plants as sources for novel antimicrobial agents. The need to expand the available pharmaceutical repertoire is underlined in several recent reports, including the 2019 Antibiotic Resistant Threat Report by the Centers for Disease Control and Prevention. This document states in the United States alone there are more than 2.8 million antibiotic-resistant infections and more than 35,000 related deaths each year. These fatal infections are most frequently caused by the 18 species of bacteria and fungi listed as current, urgent, serious, and concerning human health threats. Additionally, on a global scale, infectious diseases cause approximately 20% of all deaths each year and are the leading cause of death of children under 5 years old. Many in the medical field agree that devastating statistics like these are a consequence of entering the post-antibiotic era, a time in which the efficacies of antibiotics and other antimicrobials are unreliable. Despite the continuous popularity of herbal medicine across the globe, traditional antibiotics have previously overshadowed the exploration of plant-based products as therapeutics. However, due to the growing need for new antimicrobial agents, many scientists have now expanded their searches to include novel plant and other environmental sources. Indeed, mainstream medicine is increasingly receptive to the use of plant-derived drugs, especially those of which antimicrobial resistance is more difficult or unlikely to develop. Notably, 20% of all new approved drugs and 33% of all new small molecule approved drugs between 1981 and 2014 were botanical drugs, unaltered natural products or derivatives thereof. This abundance underscores the vast, untapped potential of plants around the world to yield desperately needed novel drugs. In fact, around only 6% of the 300,000 species of higher plants have been pharmacologically investigated. However, recent reviews have highlighted the increasingly evident antibacterial properties of various plant species and phytochemicals. Furthermore, there's an increasing amount of evidence that suggests that phytochemicals may be used in conjunction with current antimicrobials to obtain synergistic effects. For example, an early study found that crude extracts of multiple plant species shown in vitro synergistic activity with existing antibiotics when used against two multi-drug resistant enteric bacterial species. Thus, the combination of phytochemicals and antibiotics may help to combat resistance to conventional monotherapies for many diseases. As evidence for the use of natural products to treat human disease continues to accumulate, it will become increasingly important to perform in-depth safety studies on the identified extracts, compounds, and their derivatives. So far, there's a general consensus that natural products, as compared to synthetic drugs, have relatively low toxicity to mammals and less harmful effects on non-target beneficial organisms, which is yet another appealing aspect of utilizing plant species for the identification of effective pharmaceuticals. The importance of neem as a medicinal plant. Commonly known as the neem tree, is a tropical evergreen tree that's native to the Indian subcontinent. For thousands of years, neem has been recognized for its wide array of beneficial properties, including those in agriculture for pest control and in traditional medicine for various common human ailments. A. indica originally provoked worldwide interest due to its capacity as a non-toxic infection control agent for use in farming. Indeed, one of the most abundant compounds found within the neem plant is an increasingly common biopesticide. However, various parts of the neem tree have been used for millennia in traditional Indian medicine for their claimed antipyretic, anti-acid, antiparasitic, antibacterial, antiviral, anti-diabetic, contraceptive, antidermatic, anti-cancer, anti-inflammatory, antioxidant, antifungal, dental, and other healing and protective properties. Almost every part of A. indica, example the stem, bark, roots, leaves, gum, seeds, fruits, flowers, have been used as a household remedy for human illnesses. Moreover, millions of people globally use neem twigs as a source of chewing sticks for dental hygiene. More recently, the neem tree has gained attention from modern medicine and infectious disease researchers as a potential source for new antimicrobials, in addition to the application of A. indica in the fields of oncology, dentistry, dermatology, and endocrinology, among others, for reviews of some of these individual topics. Subsequent sections of this review seek to provide an overview of the most recent scientific findings that support the consideration of neem extracts and phytochemicals as antimicrobial agents. Specifically, the potential for neem and neem-related products to target pathogens that are resistant to first-line antibiotics, bacterial species that affect oral health and/or form difficult-to-eradicate biofilms, fungal infections that threaten food sources, and viral infections that have major impacts on human health are highlighted. Research in these fields is supported by a worldwide interest in neem products that stretches from ancient medicinal practices to the abundance of publications that highlight A. indica as a plant with modern pharmacological attributes. Apart from the activities highlighted herein, we'd like to point the reader to the expert review that covers the most recent evidence supporting neem as a treatment for specific human ailments and known mechanism of action of neem components therein, and specific protective qualities, and the related clinical trials are there thoroughly reported. Phytochemistry. Although nearly every part of the neem has been used for traditional medicinal purposes in India, the most widely available A. indica product on the market today is neem oil. The country of India alone produces hundreds of thousands of tons of neem oil annually, and a byproduct of neem oil production includes neem cake, which is abundantly used in agriculture around the world at less than 600 pounds per acre of farmland. Neem oil is considered a vegetable oil that is cold pressed from the fruits and seeds of neem. However, neem oil can be further processed into various types of extracts via different solvents that are then used for subsequent preclinical and clinical studies. Although various solvents can be implemented to extract different active components from plant products, most of the compounds that are thought to be responsible for the biological activities of neem can be found in extracts that are typically used in laboratories water, ethanol, methanol, chloroform, and ether. In recently published literature, methanol and ethanol extracts are those that are most commonly used for antimicrobial testing. The general biological activities of attested neem oil extracts have been attributed to the presence of many secondary plant metabolites, which include classes of compounds such as isoprenoids, terpenoids containing laminoid structures, and non-isoprenoids. The neem tree contains hundreds of compounds, phytochemicals, many of which have been found to be bioactive and to have diverse utility on their own. Out of the more than almost 300 unique compounds have been identified within the neem tree, some of the more abundant phytochemicals have already been defined as potential drugs with a wide range of biological activities. The compounds that have been most thoroughly investigated for their individual antimicrobial properties thus far are laminoids, which are compounds that typically consist of 4 6-membered rings and 1 5-membered aromatic ring, and make up 1/3 of the phytochemicals derived from the neem tree. This class of compounds also has been explored for its abundance of antioxidant activities that include nimbulide, nimbin, and nibidin, and denidin, azadirachtin. Previously established in vitro activities of these compounds and others isolated from neem seed oil have been reviewed and range from anti-inflammatory and anti-ulcer to spermicidal and anti-psoriasis. 2 recent comprehensive reviews have expertly covered the extensive phytochemistry of neem. Antimicrobial testing and safety. To test the antimicrobial activities of neem oil extracts and phytochemicals in vitro methods such as broth dilution, disc or agar diffusion, and agar overlay assays are commonly used to determine the minimum inhibitory concentration and minimum bactericidal concentration of each treatment. A few in vivo models have been implemented to more accurately reflect human infection and disease testing. These models include intraperitoneal and intravenous injection or oral and gastric administration of neem oil-related drugs in mice, rats, guinea pigs, and rabbits. These published animal studies indicate that the acute toxicity level of neem greatly depends on the plant component and solvent used to make the extract, as well as on the treatment route and species used in the model. For a comprehensive review of this topic, as an example, oral administration of the ethanolic neem leaf extract less than 2,000 milligrams per kilogram of body weight did not cause mortality in mice. Conversely, the ethanolic extract of neem stem bark given to rats at 50 to 200 milligrams per kilogram altered the biochemical markers of toxicity and may have consequential effects on organ function. In contrast, one small study showed that the glyphosate powder of an aqueous neem bark extract, given at doses of 30 to 60 milligrams twice daily for 10 weeks, had therapeutic potential in adults for controlling gastric hypersecretion and gastroesophageal and gastroduodenal ulcers. There were no obvious effects on blood parameters indicative of organ toxicity. Also, after 1 year of external exposure to neem oil, 156 adults and 110 children did not experience any major adverse effects. Of note, they showed that the wax moth larvae model is a reliable method to assess the acute in vivo toxicity of medicinal plants, in which ethanolic extracts of neem leaves and seeds had higher levels of toxicity than aqueous extracts of the same material. Although the available in vivo data will need to be further developed before neem oil extracts and phytochemicals are applied to a clinical setting, the United States Environmental Protection Agency has stated that cold-pressed neem oil should have no unreasonable adverse effects to the U.S. population and the environment. For neem-derived products, non-aqueous extracts are generally the most toxic, while unprocessed materials and pure phytochemicals from the neem tree have relatively low toxicities. Given the currently available information summarized here, an important goal of future antimicrobial testing of neem oil and its products should be the standardization of extracts and administration method. Additional, more consistent studies on this topic may allow research to draw more detailed conclusions about the potential use in a clinical setting. Antimicrobial evidence. The rising rates of antibiotic resistance for bacterial pathogens has led to the need for novel therapeutics. Thus, much of the recent work on the antimicrobial potential for neem has focused on the antibacterial properties of the plant. This area of research is supported by the traditional use of neem products for dental hygiene and the successful application of neem in the food industry. In addition to standard antibiotic resistance, the ability of pathogenic bacterial species to form biofilms has led to an increased interest in describing how these communities contribute to heightened tolerance to antibacterial substances. Although the importance of biofilm-associated infections in human disease is well recognized, few novel solutions have effectively eliminated biofilms have been developed so far. However, encouraging data suggests that neem is consistently more effective in prohibiting bacterial growth and in targeting biofilm-grown cells and many other herbal extracts, and therefore worth pursuing as a source for drug discovery. For a more comprehensive list of the antibacterial properties of neem tree, see the supplementary table. Dentistry. As described above, the use of neem twigs as dental cleaning sticks is commonplace in many countries to which it's a native species. This traditional use has translated to a growing number of studies that have tested neem products for their ability to improve dental hygiene and to prevent and treat oral diseases. Typically, these studies aim to identify an application for neem extracts as a mouthwash alternative, root canal irrigant, toothpaste, etc. Previously, the antimicrobial activity of neem against common endodontic pathogens, such as Endococcus faecalis, Staphylococcus aureus, Streptococcus mutans, and Candida albicans, have been well established. For example, a decade ago, it was shown that 7.5% aqueous neem leaf extract was able to inhibit the growth of E. faecalis, S. mutans, and C. albicans, and that MIC of an ethanol neem late extraction at 1.188%, 7.5%, and 3.75% against these three important dental pathogens, respectively. More recently, another group was able to determine that a methanolic extract of A. indica showed considerable antimicrobial activity against a 3-week-old polymicrobial dental biofilm shown on extracted human teeth, consisting of S. mutans, E. faecalis, A. aureus, and C. albicans. The result from 2 small human studies indicate that neem-based toothpaste or gel can reduce the levels of S. mutans in the mouth, and that the gel formulation can reduce plaque and gingivitis to the same degree as a chlorhexidine gel control. Additional biological relevant studies may be able to provide even more evidence for the use of neem in dentistry in combination with and as an alternative to antimicrobials already used in the field. Of note, several studies have already suggested that neem extracts have similar levels of activity as chlorhexidine and hypochlorite, typical components of oral washes, against plaque, gingivitis, and pain, and against biofilm-forming bacteria, another human pathogen that causes plaque and other biofilm-related diseases in the body. E. faecalis is just as susceptible to various neem extracts as is to chlorhexidine in vitro. The relevance of neem as an antimicrobial in dentistry, so far the most researched area, has led to many conclusions about neem extracts as compared to those from other plants used in traditional medicine. In fact, some studies suggest that neem has a greater antibacterial activity than Commiphora myra, acacia, cinnamon, salvadora, Syzygium clove, Zingiber officinale ginger, Allium sativum garlic, and Cumara longa turmeric extracts against some species of bacteria and cultured dental caries. This being said, it's important to note that certain bacterial species appear to be more susceptible to extracts from other plants in some studies. While this does not diminish the antimicrobial potential, it does underlie the importance of thoroughly taking advantage of a wide variety of antimicrobial plants and compounds that are at the disposal of modern medicine. The use of neem in the food industry, originally introduced around the world as a potent pesticide and fertilizer for agriculture, Neem has been recognized in more recent years as a safe and effective broad-spectrum antimicrobial with uses throughout the food industry that range from food production and storage to packaging and human consumption. During meat production, the presence of several species of bacteria can affect the quality and safety of product. Neem cake extract, which is a waste product from neem seed oil production, has antibacterial activities against all of these potentially pathogenic species. Additionally, Rava and Cohen found that neem leaf and bark supplements were able to successfully eliminate E. coli from cultured cow manure because this E. coli strain was isolated from an apple juice outbreak. These results may have a broad application on farms where crops and orchards frequently exist in close proximity to cattle. In the production of another source of protein for human consumption, specifically shrimp agriculture, antibiotic resistant can compromise both shrimp and human health. A thorough study on the potential use of neem in this industry included in vitro and in vivo assays that showed that aqueous neem extracts had a MIC was able to significantly increase survival of shrimp by 76% as compared to the untreated control. One of the next steps in the production of pathogen-free human food is storage and/or packaging. In the last couple of years, several groups have shown that food preservation films that are made from polyethylene or sustainable materials such as seaweed can be manufactured to incorporate neem leaf extracts, neem oil, and other plant-based products, example turmeric and curcumin. The resulting composite films are shelf-stable, block ultraviolet light, and have increased antifungal and antibacterial activities against C. albicans and a wide range of gram-negative and gram-positive organisms, including E. coli, SRS, Pseudomonas aeruginosa, and Bacillus subtilis. Furthermore, the ability of neem to prevent activity of food spoiling fungi is evident by recent reports that outline the following. The ability of neem oil to prevent the growth of grape-produced spoiling species and to inhibit the production of mycotoxin by strains of this fungus. The ability of neem leaves to prevent the production of aflatoxins and Aspergillus parasiticus during long-term storage of rice, wheat, and maize. The ability of neem seed methanol and ethanol extracts to inhibit Aspergillus flavus and A. parasiticus by 10% in the context of maize storage. The ability of multiple neem seed, bark, and leaf extracts to inhibit the growth of 3 major potato spoiling fungi. And the ability of aqueous neem leaf extract to inhibit growth of A. parasiticus as well as detoxify aflatoxin B1 and orchotoxin A. These diverse antifungal properties of neem are highlighted in supplemental table S1. Altogether, these food-related studies should encourage further investigation of the utility of indica-derived products throughout the food industry, and these products may serve as a sustainable antimicrobial alternative that can potentially improve food security via improved stable long-term storage, as well as improve human health through the elimination of food-borne pathogens. Additional antibacterial activities of neem. In several areas of antibacterial discovery, neem has been shown to be effective against many important human pathogens. Overall, a large number of studies have been published in the last decade on this topic, especially as they relate to the ever-growing number of antibiotic-resistant organisms. In this vein, several bacterial species that cause wound infections are found on the long list of antibiotic-resistant threats. Several groups have tested the antibacterial activity of neem against these species. For example, they demonstrated that methanol and chloroform neem extracts perform better than other plant extracts and better than several different antibiotics against both S. aureus and P. aeruginosa. More specifically, it has also been determined that the MIC of laminoid compounds that were isolated from neem seeds were between 32 and 128 against the P. aeruginosa and another opportunistic skin pathogen, Staphylococcus epidermidis. As a translational approach to this area of research, several studies have demonstrated that an aqueous neem leaf extract that was used to make alginate fibers for wound dressing, a nanofibrous mat embedded with neem leaf extract, and a topical gel containing a methanolic neem extract all inhibit SRS growth. In the case of polyester synthesized from neem oil to produce a nanofibrous mat, the incorporation of A. indica into this wound treatment method resulted in increased tissue regeneration in rats as compared to the control commercial cream. These results potentially have broad application for many areas of medicine in which the risk of antibiotic-resistant skin wound infection is high. Another large group of infectious bacteria that cause morbidity and mortality all over the world are the gastrointestinal pathogens, which include foodborne and diarrhea-causing organisms. Relatedly, some of the traditional uses of neem are anti-diarrheal, anti-acid, and anti-ulcer. This has led to a large body of research that has investigated the antibacterial properties of neem products against pathogens such as Salmonella, Shigella, E. coli, Listeria, and Bacillus. To summarize, the Salmonella and Shigella have been tested, which includes more than a dozen multi-drug resistant isolates from patients suffering from typhoid fever complications, are susceptible to seed, bark, and leaf extracts of neem from either ethanol, methanol, and acetone extraction. In some cases, the activity of neem extract was also found to be greater than the genicin, erythromycin, and other plants used in traditional medicine. Similarly, dried leaf, seed, and bark neem extracts in any of the three previously mentioned solvents has significant antibacterial activity against E. coli, with the methanolic extract of neem seeds demonstrating the greatest level of activity. Additionally, neem oil was as effective as ciprofloxacin against 48 tested isolates of E. coli, 14 of which were diagenic strains. Given that there are more than 250,000 cases of E. coli each year in the United States alone, associated worldwide morbidity is a huge issue for this pathogen. Indeed, resistant E. coli infections were found to be responsible for nearly a quarter of all disability-adjusted life years caused by resistant bacterial infections in the European Union in 2015. On the chronic disease spectrum of gastrointestinal illnesses, Helicobacter pylori is a pathogen that colonizes approximately 50% of the human population and causes ulcers in millions of people each year. It also causes stomach cancer in less than 1% of the people who are infected. Recently, 2 studies found that neem oil extract and a neem-associated phytochemical, nimbolide, have potent in vitro bactericidal activity against H. pylori in liquid cultures and in biofilms. Similarly, another group determined that the ethanolic neem leaf extract had activity against this species as well. Overall, studies like those described above provide strong evidence that novel plant-derived treatments like neem oil extracts and or the phytochemicals contained therein may be able to reduce the burden of the pervasive organisms like E. coli and H. pylori. Finally, there have been many studies published on the antibacterial properties of neem against a variety of diverse human pathogens. The overwhelming conclusion from the majority of these investigations is that many elements of A. indica produce extracts that have moderate to significant levels of antimicrobial activity against several pathogens. Moreover, a side-by-side comparison, the neem components often show superiority to other plants. A few exceptions include aqueous garlic extract and ethanolic green tea extract that were each shown to work better than neem extracts to kill Bacillus, Anthracis, SRS, and E. coli, respectively. Overall, studies support the notion that neem is an omnipotent plant that possesses antimicrobial activity against many bacterial pathogens. Further studies should include more standardized research approaches to test the potential for neem-derived products and individual compounds against other microbes, both in vitro and in vivo. A priority should be placed on determining the mechanisms of action of these products in order to fully understand the ideal clinical significance of Neem. Neem and biofilm forming pathogens. Biofilms are communities of bacteria composed of biofilm associated cells and extracellular polymeric substances. Components are notably recalcitrant to outside stressors, including those from the immune system and from therapeutics. Depending on the species of biofilm associated bacteria, the mechanisms of tolerance include slow diffusion, limited penetration of the drug, metabolic heterogeneity and decreased growth rates among the cells within the biofilm, and the formation of persister cells. Backed by thorough research, plants such as Neem may provide the tools needed to treat pervasive biofilm infections. To this end, there's evidence that natural products may be an ideal source of quorum sensing inhibitors, efflux pump inhibitors, and metal chelators, which are all potentially powerful anti-biofilm agents. Additionally, synergistic interactions between the many constituents found within plant extracts may provide a benefit over a single isolated ingredient. This may explain the efficacy of lower dose of herbal products like neem oil as compared to individual compounds. Significant biofilm-associated human infections are caused by species such as SRS, E. faecalis, and P. aeruginosa. There's also evidence that Neem has activity against biofilm-forming strains of some of these pathogens. For example, neem leaf ethanolic extract was found to inhibit the SRS and methicillin-resistant SRS MRSA biofilm adherence. Most recently, another group found that a petroleum ether neem extract had an MIC and MBC of 125 and 500 respectively against a strain of MRSA. In the same in vitro study, the addition of 1 milligram per milliliter of neem extract resulted in a 68.9% reduction in MRSA biofilm. 2 milligrams per milliliter of the same extract resulted in a 83.8% reduction. Additionally, they found that ripe neem seed extracts had anti-biofilm activity against SRS and Vibrio coli. The minimum biofilm inhibitory concentrations and minimum biofilm eradication concentrations for this extract were 100 and 300 respectively against SRS and 350 respectively. Furthermore, using a mouse model of V. coli infection, and others showed that administration of methanolic neem leaf extract reduced the intestinal fluid secretion, inhibited the hemorrhage in the murine, and tested without signs of a toxicity. Of note, individual Neem-associated phytochemicals, such as catechin, may have greater potential for biofilm eradication, persister cell damage, disruptor of EPS structural components, and prevention of quorum sensing. In addition, a methanolic neem oil extract killed both cells within in vitro biofilms produced by the gram-negative carcinogenic pathogens like pylori. Overall evidence suggests that neem has great potential to be used as a therapeutic for resistant bacterial infections. However, future research that utilizes animal models will be crucial to determine whether neem-derived products fit in with established antibiotic regimens and to work alone to eradicate biofilms in vivo. Antiviral evidence. Although most studies have investigated the antibacterial and antifungal potential of neem, some work has also explored the antiviral activities of neem. This topic has previously been reviewed, and to date, most publications have centered around HIV, as well as herpes, dengue, and influenza viruses. However, recent reports have included SARS-CoV-2, which was responsible for the COVID-19 pandemic. Moreover, a few groups have successfully experimented with the use of neem products against other viruses, such as Japanese encephalitis virus, hepatitis C, Coxsackie virus, and the antiviral studies have primarily focused on the ability of individual neem-associated phytochemicals to block critical processes of the viral life cycle, including cell entry and replication. Intriguingly, this means that the obtained results may describe a mechanism of action as well as identify a distinct drug candidate that could be directly modified for pharmaceutical development. Finally, and importantly, as an alternative method to control specific classes of viral and parasitic diseases, many researchers have demonstrated the ability of Neem derivatives to deter or to negatively affect the many species of insect vectors that transmit these pathogens. Neem and HIV. Human immunodeficiency virus, or HIV, is arguably one of the most devastating modern human pathogens. Since its discovery in the 1980s, there have been over 1 million new HIV infections each year. Hundreds of thousands of people still die with a subsequent acquired immune deficiency syndrome annually. Although antiretroviral therapy is well established and successful at diminishing viral load and preventing disease progression, the drugs are expensive and require lifelong treatment that is not without side effects. To this end, natural products, such as those obtained from Neem that are traditionally used for HIV-associated infections, have been explored for their potential to protect the T-cell population that is vulnerable during HIV infection, to reduce persistent immune activation during ART, and to decrease the toxicity of ART drugs. For instance, small trials have concluded that neem leaf extract given daily is safe and effective at improving the T-cell counts in HIV patients. Furthermore, neem and senna leaf extracts were given in a combination with ART. This HIV patient group had improved T-cell numbers and fewer markers of hepatic and renal toxicity than the group that was given ART alone. To address the possibility of T-cell exhaustion in HIV-infected patients, they performed an in vivo study with peripheral blood cells isolated from infected and uninfected individuals. Following exposure to enterotoxin, the lymphocytic response indicated that neem extract, but not the extracts from the two other plants, were able to down-regulate the T-cell activation in a concentration-dependent manner without affecting general